Okay, so just to move on from last week, because last week we talked about homeostasis and some key terms, including negative feedback, input, output, control center. Today we are going to go and look into excretion, because what we will do is that we will then look at the structure of the kidney. So why do we need to excrete products? Because it's one of the importance of homeostasis. So one of the physiological processes that needs to happen with the help of homeostasis is the removal of waste products. So the main excretory products of mammals include carbon dioxide, urea, and water. Right? So hopefully you remember that carbon dioxide is formed from the respiration. So when cells respire, carbon dioxide is released as the waste product. The excretory organs would be our lungs, and how it will be removed is through the exhaled air. Urea, okay, is formed from the deamination of excess amino acids. The excretory organs will be our kidneys. So kidneys are the one that's going to be excreting urea, because urea will then be removed. Okay, urea will be removed as urine. All right. And then water. So water is produced as a result of respiration in all of our cells. So the excretory organs include kidneys as part of our urine, the skin when we sweat it out, and then also our lungs as exhaled air. This is just a summary table that you can take note of to remind ourselves what are the major excretory products of mammals, how they're formed, what are the excretory organs, and how it is removed. So why is it that it's important for us to excrete, do excretion or remove any metabolic waste products? For example, in carbon dioxide. So to link this back to our AS level knowledge, you remember about the transport of carbon dioxide and what it forms when we have a lot of carbon dioxide in our blood. So essentially, any excess carbon dioxide in the blood would cause the decrease in pH, meaning that the blood will become more acidic. That's not good. Which you have already mentioned and as well as able to explain from last session, that we know that any changes in the pH, so if there is extreme changes, maybe it's too acidic or it's too alkaline, we know that it may alter the tertiary structures of the enzymes as well as other proteins, and therefore causing the enzymes or those proteins to stop working, because we're disrupting the ionic bonds that's holding this tertiary structure together. So what happens essentially is that any buildup of carbon dioxide will be detected in the brain, which is by the medulla. That would cause an increase in the breathing rates to remove these excess carbon dioxide. Right, so that's why it's important for us to remove carbon dioxide. So what we're going to focus on today is the importance of the removal of nitrogenous waste and how we excrete our nitrogenous waste product S. So we are going to look at the formation of urea. Okay, and then we're going to look at the roles of our liver. And then after that, after we learn and go through these, we will then see how the kidney will do its job in, again, removing urea as part of our urine. But before that, let's look at how urea is being formed. So mammals will excrete their waste nitrogen as urea. So that's the major form of how urea is being excreted. So if we consume more protein, we will be producing excess amino acids. So any excess amino acids will not be able to be stored in the body. So what happens is that it will undergo deamination process, where we're breaking down these excess amino acids in the liver, which produces ammonia, right? And then we will see that what happens is that the urea will then be transported to the kidneys as part of our, through our blood plasma. And then our kidney will remove the urea from our blood. Okay, it will be excreted as it is dissolved in water and as part of our urine. So what is deamination? Okay, we are going to look at that soon. So after going through deamination, okay, the waste product is ammonia. Now the problem with ammonia is that it is highly toxic. Okay, any buildup in the blood, any buildup of ammonia in our blood can cause damages to our tissue. So what happens is that the ammonia will be immediately converted to urea. So this is through the process of the ornithine cycle or the urea cycle, where we combine ammonia with carbon dioxide. So if after combining ammonia with carbon dioxide, we'll get urea, which is the main ways of how we excrete our nitrogenous product, excretory product, because urea is much less soluble and it's much less toxic comparing if we compare it to ammonia. However, other excretory products might also include creatinine. So creatinine is formed from the breakdown of creatine, which is used in the muscles, as well as the uric acid, which is formed from the breakdown of purines from the nucleotides. So this is a summary of how deamination occurs. So recapping back, I'm pretty sure you would be able to remember that amino acids is being absorbed in the ileum, and you do know that what are the uses of amino acids? Basically, it is the monomers for proteins, and proteins are important for many different functions as well. Any excess, however, any excess amino acids are taken to the liver. That amino acid will then be split, where it will undergo deamination. So deamination essentially is the removal of NH2 group and addition of hydrogen, which forms ammonia. Ammonia will then be combined with carbon dioxide to form urea. So this is via the ornithine cycle, and urea will be transported to the kidneys through the blood plasma for it to be then excreted as part of our urine. Whereas the rest of the amino acid molecule, okay, it will then be used in respiration. So it will go back into the Krebs cycle, actually, specifically the Krebs cycle. So let's do that right there. So here, you can take note, it's the Krebs cycle. And then after that, so some of them will enter the Krebs cycle. The rest would either be converted to glucose or glycogen or fat. So this is a summary of what happens to the amino acids that's consumed by mammals, right? Okay. So that's an example. So here is a self-assessment question, an example of how, okay, it's asking us to tell that, so knowing that uric acid is a nitrogenous waste product, which is produced from the breakdown of purines, what are purines and what they are used for? And the second one is explain why it is important that carbon dioxide and nitrogenous wastes are excreted and not allowed to accumulate in the body. So since we have gone through that earlier few slides, what you can do now is you try and answer the questions here, and we can discuss this when we meet the next lesson. Okay. So deamination. Urea diffuses from the liver cells into the blood plasma. So blood will then passes through the kidneys, where the urea is going to be filtered and then excreted out. Okay, so question. So this is an example of a past examination question asking us to describe how nitrogenous waste products are formed and explain why they need to be removed from the body. So this question is asking you to detail out, okay, how the different types of nitrogenous waste products are being formed. Okay. So the points that we want to see here is you to be able to explain, okay, how urea is formed. So of course, okay, nitrogenous waste products are only formed because when we consume excess amino acids. So you talk about how, okay, consuming excess amino acids, okay, what happens to these excess amino acids? So you need to start talking about how you will be removing the NH2 group and then combining it with hydrogen to form ammonia. So once ammonia is formed, ammonia is then further converted into urea by combining it with the carbon dioxide through the ornithine cycle. And that urea will be transported to the kidneys to be excreted as part of our urine. But also remember, there are other excretory products as well. So you can also mention what is creatinine and what is the uric acid as well. Okay, so a good practice would be if you could try to explain and answer this, and then we can check how you form your sentences. So through your sentences, we can see whether you've understood or if you're able to explain this process as well. Okay, so you have a reference here. Again, okay, so you can try and build your sentence ready for this. The next lesson, you can have a quick check. So structure of kidney. What is important? So students are expected to be able to identify, recognize, and label the structure of kidney. So this is a very familiar diagram. I think this is what you always see or gets asked or learn already when you were in O-levels, in your O-level studies. This is what you're familiar of. All right. So what's important is that you must be able to describe the structure of the kidney. So this is your kidney. Okay, hopefully you do know that we have two kidneys. It's located at the back. Okay. So if we actually cut through the kidney, okay, what do we see? All right, so what do we see? What does the kidney consist of? So here, okay, from A, I want you to remember where is the cortex and where is the medulla. So this is very important. No? It's important that you know where is the cortex and where is medulla. Okay, so within here, okay, within your kidney, okay, we have millions and millions and millions what known as the nephrons. So we have the nephrons. We have millions of nephrons. Now, nephrons are the most important component of your kidney. Yeah? They are the one that's doing the hard job at filtering all these waste products. All right. So once we go through the nephrons, you are required to be able to, okay, identify and label the structures. So you must be able to identify and label where is the efferent arteriole, the efferent arteriole, the glomerulus. Okay, how does the glomerulus look like? What it is? So basically the glomerulus is the knot of capillaries. The Bowman capsule, that is the one that is surrounding the glomerulus. All right, and how it looks like. There are some identifying features that we can remember there. All right, and then you are required to know the proximal and the distal convoluted tubule, the loop of Henle, and then the collecting duct. And as we go along later on, we are going to go through the structures, these different structures, and describing their functions as well. Okay. Oops, sorry. Right. So kidney. we have two kidneys at the back of our abdomen. So function, it filters waste products out of the blood. So the renal artery is the one that receives blood. Please remember that when blood enters the renal artery, it enters at very high pressure, because high pressure is needed for filtration to be more efficient, because we will see that filtration is going to be happening, right? And how there are several, how the filtration happens, and some the filtrate will form in our glomerulus later on. And the blood will leave via the renal vein, all right? And within our kidney, like what I just mentioned, it contains millions and millions of nephrons, which is the functional unit of the kidney, which is really, really important, the most important part of our kidney. The filtered waste is, so any filtered waste is released or excreted as urine, and that urine will pass from our kidney to the bladder via the ureter, which you have learned already, yeah? So any urine will pass to the bladder through the ureter, okay? And then it will be released or passes out along the urethra, so which you have already learned that during O levels. Again, very, very important. You must know where the cortex is, right? So the outer region that you can see here, this is called the cortex, okay? And then the one that you can see, so sort of like triangular structure, slightly underneath, is the medulla. Okay, please know the cortex and the medulla. So within the cortex, you will find lots of capillary network and lots and lots and lots. In fact, what they mentioned is there are millions of nephrons. So these nephrons are the functional units of our kidney. All right. So after that, you will have the central part of the kidney, which is known as the pelvis. So the pelvis is where water will be collected for our bed, okay, before it gets passed on through along the ureter. All right. So again, very important. You know where the cortex is, where the medulla is, where the pelvis is, and where ureter is. So that's from your O-level knowledge. Okay, right here, the structure of nephron. Hold on. All right, so for the structure of nephron, okay, you are required to know so you are required to be able to describe the nephrons. So essentially what you see is you will have the Bowman's capsule. See if I could zoom it in actually. Okay, so we have the Bowman's capsule. So it is characterized by this U-shaped structure here. So the Bowman's capsule is the one that's going to be, so you will have the glomerulus here right next to the Bowman's capsule. Okay, because we will see some filtration occurring, ultrafiltration occurring, and we will see the filtrate entering the Bowman's capsule later on when we look at the structures and functions of the kidney. The Bowman's capsule would then be connected to the proximal convoluted tubule. Okay, the proximal convoluted tubule will then be connected to the loop of Henle. Okay, loop of Henle. Later on, okay, you will see, and I think you have seen from the video that the loop of Henle contains so it has descending limb which goes downwards and the ascending limb which goes upwards, and then it will be connected to the distal convoluted tubule, right? The distal convoluted tubule will be connected to collecting duct, and the collecting duct will be connected then to the pelvis, where some of the water will be stored before it gets transported along the ureter. Right. So again, essentially, yes, you need to be able to describe the structure here. So my question is, okay. So my question is, do you know, okay, what does it mean by the word proximal and distal? So in anatomy, when you say something is proximal, it is close. Distal is further away. So in this case, proximal is sort of closer to the Bowman's capsule and distal is sort of further away from the Bowman's capsule. So which one comes first or closer? You can remember it's proximal. Proximity. distal distance, sort of like further away. So hopefully that could help you remember. Yeah. Right. So these are basically the notes that follows what I have just gone through earlier. So of course, if you notice here, so our proximal and distal is actually within the cortex, whereas our loop of Henle and our collecting duct is found in the medulla. Okay, and of course, we are going to look at what are their roles, which can be explained when we look at the functions of these structures later on. Why? So, what are their roles and what happens in the medulla? Okay, as well. All right. So the structure of nephrons. Okay. So we know that blood enters the kidney through the renal artery. Okay, I want you to notice. Okay, I want you to notice the diameter of the two arterioles here. So we have the afferent arteriole and we have the efferent arteriole. So tips for you to remember. It's kind of like if you want to remember which arteriole comes first, you just remember in, alphabetical order. So A comes first before E. The second is remember that the afferent arteriole has larger diameter compared to the efferent arteriole. So this is very important because this would allow blood enter, okay, the glomerulus to enter the kidney at high pressure. Okay, this high pressure is required to allow for filtration to occur, Right. So as blood enters the renal artery at high pressure, okay, it will be then connected to glomerulus, which is a knot of capillaries, right? And then it will then be joined by the efferent arteriole as it brings the filtrate out. And then you will have the Bowman's capsule, the U-shape, so or the cup-shaped structure of the nephron here, right? And then again, because this is a zoom in, so we can just go back and describe again. So the Bowman's capsule will then be connected to proximal convoluted tubule, and it will then be connected to the loop of Henle, which is the descending because it goes down, and the ascending because it goes up. This loop of Henle, if you notice, it's in the medulla, and then it will be joined back with the distal convoluted tubule in the cortex. Okay. And then we will see that it is then connected to collecting duct and eventually the pelvis. So this collecting duct and pelvis is in the medulla. Okay, as part of your syllabus, and this is a past examination question as well, you are required to be able to describe the structure of a kidney nephron and its associated blood vessels. So basically, we have seen the two blood vessels there. We see the afferent and the efferent, so you need to be talking about that, and you need to be talking about all the structures that we have seen earlier, which is the Bowman's capsule, the proximal and the distal convoluted tubule, the loop of Henle, and the collecting duct. All right, so again, so what you can do is you could probably prepare your answer. We can have a look and check it for you. I'm ready for our by next lesson. Okay, so for next lesson or next week, we are going to look at the kidney function, how the kidney will make urine. So we're going to look at ultrafiltration, and then we are going to look at selective reabsorption. So we're going to look at ultrafiltration in the Bowman's capsule, the glomerulus, and then we're going to look at selective reabsorption when we're looking at the proximal convoluted tubule. So this is the plan for next. So again, hopefully if you have any questions, do prepare them next week during the next session, and hopefully you have you can complete the self-assessment questions in a past paper. Okay, I know that I have given you the mark scheme there, but what's most important is if you are able to form your sentences, that's one way of how we can check as well. All right, So that's it for the first half of our lesson. Okay.